Unit 7, Lesson 5. Here's a picture of DNA, which you already know and recognize. Here's a picture of RNA, which we haven't really seen before. What similarities and differences do you notice? You can write them here. That's a text box for you. You can see in both strands we have cytosine, guanine, adenine, and then there's a difference at the bottom. This side has thymine, this side has uracil. Take note of the shape. You can write about what they do have in common or how they're different or both. In DNA pairing, we've been calling those nucleotides chemically specific. They're also referred to as complementary. Just like complementary angles in math, both angles add up to 90 degrees, or complementary colors on the color wheel are opposite of each other. So adenine and thymine are complementary. Cytosine and guanine, those are the ones that pair together. RNA is also complementary. Again, adenine is there, but instead of thymine, there's something called uracil, which will always bond with adenine. And then cytosine and guanine stay the same. So the difference between DNA and RNA, there is no thymine in RNA. Instead, it's a molecule called uracil. So I like to remember this as say goodbye to the thigh, like a bad workout video. Here's a brief and really basic, simple overview of how RNA works for DNA to make new proteins. In a little bit, you'll have a much more in-depth and fun way of seeing how this works. But for now, the first piece is called mRNA, and this is the messenger. That's what the M stands for. So messenger RNA goes into the nucleus and gets the code from the DNA to hand it to the next, quote, guy in line. It's not a guy. It's a strand of RNA. This step is called transcription. So if you think about scribing for someone, what are you doing? You're writing something down, right? So this RNA is the very first step. It's transcribing the information from the original DNA strand. We'll talk about why it has to do that in a moment. mRNA then passes the code that it wrote down pretty much to tRNA. That T stands for transfer. So the tRNA interprets the code from the mRNA and then gives that information to the next, quote, guy in line. Guess what? It's a different type of RNA, which can then turn it into amino acids. This tRNA, the transfer step, is called translation, and we'll talk about what it's translating in a moment. The last type of RNA is called rRNA. This actually stands for ribosomal because a ribosome is made out of this kind of RNA. So the ribosome interprets the code that came from the two previous steps of the RNA and reads those 3 letters that we did the other day in class with a chart. It doesn't need a chart. It does this on its own, but we use this chart to interpret what that ribosome does. When we were converting these 3 letters into the different amino acids, we were acting like ribosomes. So that's what the rRNA does. And then those little amino acids get spit out the other side of the ribosome and build into different shapes and sizes of proteins so that you can have different cells and functions. Go ahead and enjoy this interpretive vision of how RNA works. I think you'll recognize the actors. Hello? No, I said send them through the membrane. Yes, the cell membrane. Okay, thanks, bye. You can just take it. Hey, so I just take whatever I want? No, you can't take it out of the room, but you can take it. I'm good to go? Okay. No, it can't leave the room. It's too precious, okay? Just copy it down, take it, bye. Okay. But Miss Gilman, why was the code backwards? So see how RNA is one single strand? RNA matches up with only one side of the DNA, the complementary base pairs. The DNA gets unzipped by an enzyme that leaves those base pairs free. The RNA comes in and matches up to it. But since it's the opposite side, the first strand of RNA is kind of like a mirror image. That's why the second strand of RNA, that tRNA, has to flip the code before it gets sent to the ribosome to make the protein so it matches its original DNA. By now you've seen how it can be kind of tricky to get the code right if you're transferring the information from a DNA strand through the steps that it takes to become a protein eventually. So there's a lot of room for error. And we call those little tiny errors mutations. So some traits can be the result of a change in the DNA in the first place, or it could be a transferring or copying error from the RNA that ends up as a different gene or chromosome. Mutations can cause a cell to produce an incorrect protein during protein synthesis, because if you give the wrong instructions, you're going to get the wrong result. Go ahead and answer on your own. What do you think will be the result of a mutation? Will the result of that mutation be passed to offspring? Why or why not? Question 2 is a bit of a challenge. We haven't really talked about how we pass traits to offspring specifically beyond fertilization. We'll get into that more next week. The first type of mutation we'll talk about today is insertion. So I like this GIF because it can show how the DNA is inserting itself as well. An extra base pair is added to the DNA. So here's our original sequence. I'd like you to write down below what happened there that's different. How would you describe this to somebody else? So what's the difference between our original sequence up here and the sequence below? Write it there in your own words. Then we can have a deletion. So one or more base pair can be removed from the DNA. Take a look at our original sequence here, our deletion happening down there with the arrow, and then, the deletion in this picture. And write in your own words what the difference is below. The third kind of mutation we'll talk about today is called substitution. Just like it goes in as a goldfish cracker and ends up as a fish in the GIF, a base pair can be substituted or swapped out for another in the DNA. So here's our original sequence. This could be one or two bases. This could be a whole long gene or segment of the DNA, but you can see what's happening there. Go ahead and write in your own words how you would describe what's going on. Sometimes mutations can be harmful. Sometimes they can be helpful. And sometimes they can be totally neutral. So take a look at these kangaroos. They both live in the same wildlife preserve in Australia. A mutation causes this kangaroo to lack a certain pigment. This is a regular kangaroo. Both of these live within a few feet of each other in this wildlife preserve. Do you think this mutation is harmful to this kangaroo? Why or why not? Write it down here. I'll give you a hint. There are lots of right answers to this question as long as you explain your reasoning. So for a bit more challenge, go ahead and classify these different mutations as harmful, helpful, or both or neither in this Venn diagram. You can drag these, they're individual word boxes. So think about, back in the day, a hunter-gatherer who might have been colorblind. Think about what you need to do as a hunter-gatherer. What are you looking for? Is the berry red? Is it green? Does it matter? Go ahead and drag that into there. People native to the Himalayan mountain range who have higher hemoglobin counts. Remember, hemoglobin can help carry oxygen through our blood. We talked about that the other day. So is the mutation where you can carry more oxygen at a high altitude, is that helpful, harmful, both, or neither? A white kangaroo in the wild, a white kangaroo in the zoo, think about their settings. Being colorblind today in New York City, is it harmful, helpful, both, neither? And lastly, this one's a bit of a challenge. Bacteria that is resistant to antibiotics. Bacteria that is resistant to antibiotics. So when you take antibiotics for a bacterial infection, some bacteria will not get treated by that because they're resistant. So that means they have some sort of gene or mutation that allows them to not die when they face those antibiotics. Think about that from your perspective, but also think about it from the bacteria's perspective. So is that harmful, helpful, both, or neither? Go ahead and make your decisions. All right, that is the end of Lesson 5. We will go over these tomorrow in class. Bye!